Welcome to the deep dive. We're really glad you're here with us today.
Absolutely. It's great to have you listening.
Okay, so let's unpack this. We have it's a massive mission ahead of us today.
A very big one.
Yeah. We're taking a foundational text, chapter 14 of the biology curriculum, Respiration in Plants, and we're going to completely deconstruct the cellular machinery of respiration.
Right. We are charting out the entire biochemical pathway, basically from start to finish.
Exactly. I've been poring over these notes, mapping out the molecules, and I just had this massive breakthrough in understanding how this invisible microscopic world actually pieces together to keep things alive.
It's a great feeling when that finally clicks.
It really is. But to make sure we don't just get lost in the weeds of biology here, I'm joined by someone who has spent over 15 years analyzing the minds of examiners.
That's right.
Guiding students through these incredibly competitive medical and biology entrance exams like Neet.
Yeah, I've seen it all. Every mistake, every trap.
So we're going to figure out what happens inside these cells, why the system is designed this way, and exactly how they're going to test you on it.
It is fantastic to dive into this material. Respiration is, it's one of those topics that is incredibly easy to misunderstand right out of the gate.
Oh, for sure. Yeah. I definitely did it first.
I see it every single year. Students try to brute force memorize the cycles, the enzymes, the carbon counts.
Just rote memorization.
Exactly. But they completely miss the magnificent logic of the system.
The actual engine.
Right. In my years of mentoring, the students who truly excel, the ones who hit the top percentiles, they're the ones who stop seeing these biochemical pathways as a punishing list of unpronounceable names.
Yeah.
Instead, they see it as an elegant, highly dynamic engine. So our goal today is to build that intuition for you.
We want you to walk into that exam hall, or just walk away from this conversation, not just carrying a basket of memorized facts, but with absolute crystal clear clarity on how the engine runs. So I want to start with a question that bothered me for a long time when I first cracked open this text. It's right there at the beginning.
Okay, lay it on me.
Do plants actually breathe? Because if we look at a plant, there are no lungs.
Right, no lungs.
There's no diaphragm. You never see a giant oak tree, panting after a windy day.
No, you certainly don't.
So how does a biological entity of that massive size survive without a centralized breathing apparatus?
To answer that, we have to redefine what breathing and respiration actually mean. We got to strip away our animal-centric bias.
Okay.
Let's cut straight to the essence. Macroscopic breathing, inhaling oxygen, and exhaling carbon dioxide is just a delivery and waste removal mechanism.
It's just logistics.
Exactly. At its absolute core, respiration is something entirely different. It is the biochemical process of breaking the carbon-carbon bonds of complex macromolecules, what we just call food, through the process of oxidation, specifically to release energy.
So we're really talking about tearing apart molecules.
Tearing them apart for power.
The text points out that all the energy required for life processes, whether that's a plant pushing a root through solid soil, transporting water hundreds of feet up a trunk, or reproducing, it all comes from the oxidation of these macromolecules.
Yes.
And ultimately, every single bit of that food originates from photosynthesis. Plants trap solar energy, convert it into chemical energy, and lock it away in the bonds of carbohydrates like glucose, sucrose, and starch.
But trapping it is only half the battle. They have to get that energy back out when they need it.
They do, and they face a massive physical problem in how they extract it.
Think about the physics of energy release for a second. If a cell were to just burn a molecule of glucose all at once, in a single chemical step, the same way you might burn a marshmallow on a campfire, it would be a total disaster.
An explosion, basically.
I always frame this for my students using a financial analogy. Imagine trying to buy a single cup of coffee, but the only currency you have in your pocket is a solid 10-pound gold bar.
Yeah, the barista is definitely not going to be able to make change for a 10-pound gold bar.
Exactly. It is far too much value concentrated in one single, unusable unit. If a cell completely combusted a glucose molecule in one rapid step, producing carbon dioxide and water instantaneously, almost all of that massive amount of energy would be released at once in the form of heat.
And heat is bad for a cell.
It would literally boil its own internal fluids and cook itself. It cannot handle the gold bar.
Which means the cell needs a strategy to break that gold bar down into pocket change.
Right.
So instead of an explosive release of heat, the plant cell catabolizes or breaks down the glucose molecule in a highly controlled agonizingly slow series of stepwise reactions.
It uses specific enzymes to chip away at the molecule bit by bit.
It's like taking that massive gold bar to a highly specialized bank, melting it down, and slowly minting it into millions of usable tiny copper coins.
I love that.
And those coins, the actual energy currency of the cell, are molecules of ATP.
ATP is the universal pocket change of life. The golden rule of cellular respiration is that the energy released from breaking those carbon bonds is trapped as chemical energy in the form of ATP.
And then, that ATP can be broken down whenever and wherever the cell needs a tiny, safe burst of energy to do work.
Furthermore, by dismantling the glucose slowly, the cell produces a variety of smaller carbon skeletons.
Like little leftover pieces.
Right, and these intermediate pieces aren't just burned, they are pulled out of the pathway and used as building blocks to construct other necessary molecules. It is a masterclass in recycling and efficiency.
Okay, so if respiration is happening constantly at this microscopic cellular level, producing ATP, we still have to loop back to the breathing paradox, the logistics of gas exchange.
Let's get into it.
Because to do this efficiently, the cells still require oxygen, and they still produce carbon dioxide as waste. So without a specialized respiratory system, without blood to carry gases around, how does a massive thick-trunk tree actually orchestrate this?
The curriculum outlines 3 brilliant evolutionary strategies that plants use to solve this lungless survival problem. First, there is radical decentralization.
Meaning everyone fends for themselves.
Exactly. Every single part of the plant takes care of its own gas exchange needs. Leaves have microscopic pores called stomata. Stems have specialized openings called lenticels.
Okay.
There is almost zero transport of gases from one part of the plant to another. The leaf doesn't rely on the roots for oxygen, and the roots don't rely on the leaves.
So it's an entirely localized economy.
Yes.
That leads into the second reason the text provides. Plants simply do not present great demands for gas exchange compared to animals. Roots, stems, and leaves respire at rates far, far lower than you or I do.
Much lower. The only time there is a massive volume of gas being furiously exchanged is during photosynthesis in the middle of the day.
But during that time, each leaf is perfectly adapted to handle its own local needs.
Which brings us to the third and perhaps most elegant reason, the utter self-sufficiency of the photosynthesizing leaf.
How so?
During the day, availability of oxygen is not a problem for these cells because oxygen is being continuously released directly inside the cell itself as a byproduct of photosynthesis.
Oh, it is a closed-loop supply chain.
Precisely. The chloroplasts are generating the very oxygen the mitochondria need to burn the glucose.
But what about the areas of the plant that don't see the sun? Like the thick, woody stems or the roots buried deep underground. That's the part that used to trip me up. How does oxygen get to the center of a tree trunk?
Right, the solid wood problem.
The text explains that the actual physical distance gases must diffuse, even in the largest, bulkiest plants, is remarkably small.
This requires us to visualize the interior architecture of a tree. When you look at a massive oak, you assume the entire thing is a giant block of living, breathing tissue.
Yeah, you just assume it's all alive.
It isn't. The actual living cells in a thick, woody stem are organized in very thin layers right beneath the bark. The entire deep interior of the wood, the heartwood, is entirely dead tissue.
Wow.
It is there solely for structural mechanical support. It does not respire, so it requires no oxygen.
And for those living layers, if we bridge this back to foundational plant anatomy, we have to think about parenchyma cells.
Yes, the parenchyma.
The text explicitly points out that the loose, airy packing of parenchyma cells in leaves, stems, and roots provides a vast, interconnected network of air spaces. It is like a sponge.
That structural design ensures that almost every single living cell in a plant has at least a part of its surface membrane in direct physical contact with the air. They don't need lungs because they are practically bathing in the atmosphere.
It is an architectural marvel. But establishing how plants get oxygen raises an incredibly important evolutionary question, and it sets the stage for the very first biochemical pathway we need to master.
The origins of it all.
We have established that the most efficient, complete combustion of glucose requires oxygen. But if we wind the clock back 3.5 billion years, the early Earth atmosphere contained ammonia, methane, and water vapor. But it entirely lacked free gaseous oxygen.
This is a huge conceptual leap for students.
This was my first massive realization when going through the source material. The very first single-celled organisms on this planet evolved and thrived in an atmosphere with zero free oxygen.
They had to solve the energy problem using only the chemicals available.
And because evolution builds on what works, every single living organism alive today, whether it's an oak tree, a human reading a textbook, or a bacteria deep in a thermal vent, still retains that exact same ancient enzymatic machinery to partially break down glucose without using a single molecule of oxygen.
It is a profound realization. Life engineered a solution to extract energy long before oxygen was biologically available, and that solution was so perfectly functional that evolution never discarded it.
It just kept it.
It simply built more efficient systems on top of it later. This universal, ancient first step of glucose breakdown is called glycolysis.
Let's transition fully into glycolysis.
Yeah.
If we are building a mental map for you listening, I want you to imagine the cell as a massive, bustling corporate headquarters.
Okay, I like this.
The nucleus is the locked executive suite where the DNA blueprints are kept. The mitochondria, which we will visit later, are the heavily guarded VIP energy vault.
Then a cytoplasm.
The cytoplasm, the fluid filling the cell, that is the main lobby. And glycolysis happens right there, out in the open, in the lobby. Every single molecule of glucose that enters the cell must pass through this lobby and undergo glycolysis before it is allowed to go anywhere else.
The lobby analogy works perfectly because it highlights the accessibility of the process. Let's examine the name itself. Glycolysis is derived from the Greek words glykos, meaning sugar, and lysis, meaning splitting. Sugar splitting.
Makes sense.
And for those preparing for exams, you will very frequently see this referred to by a specific acronym, the EMP pathway.
The EMP pathway, where does that come from?
It is named after the three scientists who painstakingly discovered and mapped out this entire reaction scheme: Gustav Emden, Otto Meyerhoff, and J. Parnas.
Emden, Meyerhoff, Parnas.
You must associate the EMP pathway directly with glycolysis. They are interchangeable terms in the mind of the examiner.
Let's do the step-by-step breakdown. In a plant, where does the sugar originally come from to enter this lobby? The text tells us that glucose is primarily derived from sucrose, which is the end product of photosynthesis, or it is mobilized from storage carbohydrates like starch.
This is where we hit our first major high-yield focus area. Listen closely to this mechanism. Sucrose is a disaccharide, meaning it is too complex to enter glycolysis directly.
It's too big.
Exactly. It must be cleaved into its two simpler components, glucose and fructose. There is a specific enzyme responsible for this. Examiners love to test this exact step because it bridges photosynthesis and respiration.
Looking at the pathway, the enzyme that takes sucrose and splits it is called invertase.
Yes, here's what you must remember. Invertase is the key that unlocks sucrose. Once invertase acts, you are left with one molecule of glucose and one molecule of fructose. These two monosaccharides are now ready to readily enter the glycolytic pathway.
Let's track the glucose molecule. We know it is a 6-carbon sugar. Glycolysis is a long chain of 10 distinct chemical reactions, tightly controlled by different enzymes.
10 steps.
But we don't need to get bogged down in memorizing every single chemical intermediate. We need to track the money. We need to focus on where the energy is invested, where the molecule is broken, and where the energy is finally harvested.
Follow the ATP.
Right. The whole process starts with what is called the investment phase.
Think back to our gold bar analogy. The cell wants to break down the gold bar to get ATP. But paradoxically, to initiate this entire breakdown process, the cell actually has to spend some of its own hard-earned ATP.
It takes money to make money.
It literally takes money to make money. The glucose molecule is stable. It doesn't just fall apart on its own. It needs to be destabilized.
How does it do that? The text says glucose is phosphorylated to give rise to glucose 6-phosphate, and this is driven by the enzyme hexokinase. Can we explain phosphorylation conceptually? What is actually happening to the glucose?
Think of phosphorylation as slapping a giant, bulky, highly negatively charged sticky note onto the glucose molecule. The ATP molecule gives up one of its phosphate groups and attaches it to the 6th carbon of the glucose.
Okay, so what does that do?
Two things happen here. First, glucose is a neutral molecule that can easily slip in and out of the cell membrane. But once you attach that negative phosphate group, it is trapped. It cannot leave the lobby.
So it locks it in.
Second, that phosphate group makes the whole molecular structure unstable and highly reactive. But doing this costs the cell one molecule of ATP.
So we are down one ATP. Our glucose is now trapped and energized as glucose 6-phosphate. What happens next? The text says it isomerizes into fructose 6-phosphate. Isomerization, that just means rearranging the furniture, right?
Precisely. The molecule doesn't gain or lose any atoms. The enzyme simply shifts the existing atoms around, changing the chemical shape from a six-sided ring to a five-sided ring, creating fructose 6-phosphate. It is a necessary structural adjustment for the steps that follow.
And right after rearranging the furniture, we hit the second investment. The cell spins yet another molecule of ATP to attach a second phosphate group, turning fructose 6-phosphate into fructose 1,6-species phosphate.
So, take stock of where we are. We are deep in the cytoplasm lobby. We have taken a stable 6-carbon sugar, trapped it, rearranged it, and aggressively destabilized it by forcing two bulky phosphate groups onto it. And the cell is currently in energy debt. We are down 2 ATP molecules.
Now comes the dramatic part. The Deconstructing and Glycolysis, the splitting phase. This highly unstable, doubly phosphorylated 6-carbon sugar is literally ripped in half. It splits into two distinct 3-carbon molecules.
Those two resulting 3-carbon molecules are dihydroxyacetone phosphate, which we abbreviate as DHAP, and 3-phosphoglyceraldehyde, which is famously known as PGAL.
DHAP and PGA.
Now, DHAP and PGAL are isomers. They can quickly interconvert back and forth, but for the pathway to move forward, it only accepts PGL. So the DHAP rapidly converts itself into PGA.
Which means from our original single 6-carbon glucose, we now have two identical molecules of 3-carbon PGL moving forward. The text emphasizes this heavily. From this exact point onward, everything happens twice. Every reaction, every product, every energy yield must be multiplied by 2 for every original glucose molecule.
That multiplication is the most common place students drop points on exams. They calculate the yield for 1 PGL and forget to double it. So we have 2 PGL molecules, and we are entering the payoff phase. This is where the cell finally starts getting a return on its agonizing chemical investment.
The first step of the payoff phase involves PGL being converted into 1,4-bisphosphoglycerate, or BPGA.
We must pause here because this is a massive exam trap. This specific reaction, the conversion of PGL to BPGA, is arguably the most heavily scrutinized step in all of glycolysis by examiners.
Really? Why?
Because it is the exact moment where oxidation occurs.
What exactly is happening chemically during this oxidation? The text says 2 redox equivalents are removed.
Redox simply means the transfer of electrons. Oxidation is the loss of electrons. In this step, the enzyme reaches in and rips away 2 hydrogen atoms from the PGO molecule. Those hydrogen atoms carry high-energy electrons. But the cell can't just let high-energy electrons loose in the cytoplasm. They are incredibly destructive. They need a safe carrier.
So what catches them?
A specialized carrier molecule called NAD+ swoops in, catches those 2 hydrogen atoms with their electrons, and becomes reduced to NADH plus H plus a D.
This NADH molecule keeps coming up. I see it generated here, and I know it's going to be huge later. How should we visualize NADH?
Think of NADH as a high-value casino chip. It represents a massive amount of stored energy, but you cannot spend it down here in the lobby. The enzymes that power the cell only accept ATP coins.
You can't put a casino chip in a vending machine.
Exactly. You have to hold on to this NADH casino chip and take it to the cashier's desk later on to cash it in for ATP. But for now, here's what you must remember for the exam. You need to know exactly where this casino chip is minted.
The location matters.
If you look at past papers, examiners will describe a reaction and ask, at which specific step is NADH formed in the EMP pathway? The answer is the conversion of PGAL to BPGA.
Got it. PGAL to BPGA yields the NADH casino chip. And because we have 2 PGLs, we just made 2 NADHs.
Right.
Moving down the chain, the BPGA is then converted to 3-phosphoglyceric acid, or PGA. And finally, boom, we have an energy yielding process. The energy released from this chemical shift is so immense that it is trapped by physically forcing a phosphate group onto an ADP molecule, creating ATP.
This is direct ATP synthesis. And again, keeping our accounting straight. Since we have 2 molecules of BPGA going through this step simultaneously, we just synthesized 2 molecules of ATP.
Which means we have officially paid back our initial debt. We spent 2 at the start, we just made 2. We were breaking even.
Breaking even.
The pathway continues through a few more minor intermediate steps. PGA turns into 2-phosphoglycerate, then into phosphonyl pyruvate, or PBP.
And then we reach the final reaction of glycolysis, the conversion of PP into our final end product, pyruvic acid. This reaction releases another massive burst of energy, yielding another molecule of ATP per 3-carbon chain.
So that's another 2 ATPs produced. Let's do the final math, because this is where the textbook questions always aim. The text asks us to calculate how many ATP molecules are directly synthesized in this pathway from 1 glucose molecule.
Here is the unshakable balance sheet you must carry in your head. You invested 2 ATP in the preparatory phase. You directly synthesized 4 ATP in the payoff phase. Therefore, the net gain of ATP in glycolysis is exactly 2 molecules of ATP.
Net 2 ATP. We started with the 6-carbon glucose, and the final physical products sitting out in our cytoplasm lobby are 2 molecules of a 3-carbon compound called pyruvic acid, along with 2 ATP and 2 NADH.
Perfect. The glucose has undergone partial oxidation. Crucially, not a single molecule of oxygen has been used yet. This brings us to a major crossroads in cellular metabolism.
We're entering part 3, the crossroads. We have this pyruvic acid just sitting in the lobby. What happens to it? The text makes it incredibly clear that its metabolic fate depends entirely on 2 defining factors: what the cell currently needs and whether or not oxygen is present.
There are 3 major avenues a cell can take to handle this pyruvic acid: lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Let's explore the anaerobic paths first. What happens when there is no oxygen?
Fermentation.
Collectively, we call this fermentation. It occurs in prokaryotes, unicellular eukaryotes like yeast, and surprisingly, even in our own human bodies under specific stress conditions.
Let's unpack alcoholic fermentation first. This is the classic pathway utilized by yeast. When a yeast cell finds itself in an anaerobic environment, say sealed in a vat of grape juice, it takes that pyruvic acid and converts it into carbon dioxide and ethanol.
And here's where the examiner sets the trap. They will ask you to identify the precise enzymes orchestrating this conversion. You cannot afford to mix them up. The enzymes are pyruvic acid decarboxylase and alcohol dehydrogenase.
Let's break down those names so we don't just memorize them blindly. Decarboxylase. That means it's removing a carboxyl group, which comes off as carbon dioxide gas. That's what makes bread rise and beer bubbly. And dehydrogenase. That means it's removing hydrogen, or in this case, facilitating a redox reaction to form the alcohol.
Exactly right. Pyruvic acid decarboxylase and alcohol dehydrogenase. Now let's look at the other anaerobic path: lactic acid fermentation. The text notes this happens in certain bacteria, but most relevantly for us, it happens in animal cells, like our skeletal muscles during intense exercise.
Right. If you are sprinting as fast as you can, your muscle cells are burning through ATP faster than your heart, and lungs can pump oxygenated blood to them. The muscles literally become temporarily anaerobic.
They are starved for oxygen.
So the pyruvic acid can't go to the mitochondria. Instead, it is reduced directly into lactic acid.
The enzyme mediating this is lactate dehydrogenase, and the physical result of this is something everyone has experienced: that intense burning sensation in your muscles during a heavy workout.
Yeah, the burn.
That is the localized drop in pH caused by the rapid accumulation of lactic acid. Your body eventually has to transport that lactic acid to the liver to be converted back into usable glucose, a process known as the Cori cycle.
Now, here is a piece of the biological puzzle that absolutely blew my mind when I finally connected the dots. In both of these fermentation pathways, whether making ethanol or making lactic acid, the cell is actively using up the NADH casino chips it produced earlier in glycolysis.
Yes, it is.
It is taking the NADH and reoxidizing it back into empty NAD+ carriers. Why is the cell throwing away its high-energy chips? Why is this reset so crucial?
This is the exact concept that separates the students who just memorize from the true masters of the subject. Why does fermentation even exist? It is not because a yeast cell actively desires to make ethanol, or because a muscle cell wants to fill itself with burning lactic acid.
It's not a goal.
No, fermentation is a desperate act of cellular survival.
Because of the lobby filling up.
Exactly. Think back to the middle of glycolysis. To turn PGAL into BPGA, the cell absolutely, non-negotiably requires a constant supply of empty NAD+ carriers to catch the electrons. It is a vital raw material.
Right, without it can't move forward. Now, normally, NADH goes to the mitochondria, drops off its electrons with oxygen, and returns as empty NAD+ Wicca. But if there is no oxygen, the mitochondria shut their doors. The NADH can't be cashed in.
So the cytoplasm lobby just fills up with millions of full NADH chips, and all the empty NAD+ carriers are completely depleted.
And if there is no empty NAD+ available, the PGA enzyme stops working. Glycolysis grinds to a sudden complete halt. If glycolysis stops, the cell can't even get its meager net 2 ATP. Without ATP, the cell's ion pumps fail, its membrane depolarizes, and it dies within minutes.
Oh, wow. So fermentation is just a desperate mechanism to dump those electrons onto pyruvic acid, turning it into waste products like lactic acid or ethanol, solely to empty out the carrier and regenerate NAD plus SWIG.
That's it.
It's a waste management program. By turning NADH back into NAD plus SWIG, glycolysis can keep churning, allowing the cell to eke out just enough ATP to survive the oxygen drought. That is precisely it. It is a survival mechanism. But the text is careful to point out how incredibly inefficient this stopgap measure is. Less than 7% of the total potential energy locked in a glucose molecule is released during fermentation.
7%. That's terrible.
And not even all of that 7% is trapped as high energy bonds of ATP. Most of it is wasted as heat.
And it is incredibly hazardous, too. The products themselves are literal toxins to the cell, acid and alcohol. The text gives a brilliant specific example with yeast. Yeasts will literally poison themselves to death when the concentration of alcohol in their environment reaches about 13%.
Which naturally brings up a fascinating open inquiry posed by the text. If yeast naturally dies when the alcohol concentration hits 13%, how do we possibly produce alcoholic beverages with much higher concentrations, like a 40% whiskey or a 50% vodka?
Right, because biology has a hard limit, and that limit is self-toxicity. The yeast simply cannot survive to ferment it further.
So what's the answer?
The answer, though the text leaves it for the student to ponder, is that biology can't do it. It requires human physical intervention. We have to use the physical process of distillation. We heat the 13% mixture, evaporate the alcohol because it has a lower boiling point than water, and then condense it into a much stronger, concentrated form.
It perfectly illustrates the limitations of anaerobic respiration. It is a dead end for complex life. It is purely a stopgap. So what is the process by which organisms can carry out the complete, safe oxidation of glucose and extract all that remaining 93% of stored energy?
That requires oxygen, and it requires leaving the lobby. We are entering part 4, aerobic respiration and the Krebs cycle. For true aerobic respiration to take place, that final product of glycolysis, the pyruvic acid, must be physically transported out of the cytoplasm and deep into the mitochondria.
The mitochondria, the powerhouse of the cell. But it is not a simple open door. The pyruvate cannot just walk into the Krebs cycle. It must undergo a preparatory step, which the text formally calls oxidative decarboxylation.
The link reaction.
We commonly refer to this as the link reaction because it links the lobby to the VIP room.
And this reaction takes place specifically in the mitochondrial matrix, the dense, jelly-like fluid filling the inner chamber of the mitochondria.
It is catalyzed by a massive, complex piece of molecular machinery called the pyruvic dehydrogenase complex.
Okay, let's set up the examiner trap here because I know they love testing the intricacies of this link reaction. We have pyruvic acid reacting with an empty NAD plus carrier and a molecule called coenzyme A.
Yes.
The enzyme mashes them together to form acetyl-CoA. It mints a new NADH casino chip, and it rips off a carbon which floats away as CO2. But there is a silent partner in this reaction that students always overlook.
Yes, there is. Here is what you must remember for the exam. The enzyme pyruvic dehydrogenase absolutely requires a specific metallic ion cofactor to function properly. That cofactor is the magnesium ion, Mg2 plus D.
Why does it need a metal ion?
Because the enzymes and the phosphate groups involved are highly negatively charged, the positive magnesium ion acts like an anchor, stabilizing the negative charges so the chemical reaction can actually occur. I cannot tell you how many times I have seen a multiple-choice question asking for the necessary components of the link reaction, and the distractors will maliciously swap out Mg2 plus for manganese or calcium.
Manganese, calcium, they'll try to trick you.
You must spot the Mg2 plus bend.
Magnesium ion,
Yeah.
locked in. So keeping our molecular accounting straight, from our original 2 pyruvates that enter the mitochondria, this link reaction produces 2 molecules of acetyl-CoA, mints 2 new molecules of NADH, and releases 2 molecules of CO2. We are slowly, methodically dismantling the sugar.
And that acetyl-CoA is the golden ticket. It is a highly reactive 2-carbon molecule, and it now enters a sprawling cyclic pathway. Officially, it is the tricarboxylic acid cycle, but it is universally referred to as the Krebs cycle, named after the brilliant biochemist Hans Krebs, who first elucidated its steps in the 1930s.
The Krebs cycle, let's walk through the mechanics of it. It doesn't start with a destruction, it actually starts with a condensation. The 2-carbon acetyl group from acetyl-CoA is joined together with a resident 4-carbon compound called oxaloacetic acid, or OAA, and a molecule of water.
And what is the product of that union?
A 2-carbon piece plus a 4-carbon piece gives us a brand new 6-carbon molecule, citric acid. And the enzyme that forces them together is appropriately named citrate synthase. During this fusion, the coenzyme A carrier is released, so it can go back and grab another acetyl group from the link reaction.
And because this first stable product, citric acid, is a molecule that contains exactly 3 carboxyl chemical groups, that is why the entire pathway is technically named the tricarboxylic acid cycle.
That makes perfect sense.
Yeah.
Now, the rest of the cycle is essentially a highly controlled carbon countdown. We need to completely oxidize this molecule, tearing it apart to harvest electrons. The citrate undergoes a quick isomerization, rearranging itself into isocitrate, and then we hit 2 successive violent steps of decarboxylation.
Break down decarboxylation conceptually for the listener.
Decarboxylation means the enzyme uses a chemical cleaver to chop off a carbon atom, which immediately bonds with oxygen and floats away as CO2 waste gas. So let's track the numbers. We start with our 6-carbon citric acid. We chop off a CO2. We're now left with a 5-carbon compound, which the text identifies as alpha-ketogluteric acid.
Alpha-ketogluteric acid, yes.
And right at the moment the bond breaks, we harvest the high-energy electrons, reducing an NAD+ into an NADH casino chip.
Precisely. And then the engine turns again. The 5-carbon alpha-ketogluteric acid is subjected to a second decarboxylation. It loses another CO2, shrinking down into a 4-carbon compound called Succinyl-CoA. And once again, the breaking of that bond releases electrons, generating yet another NADH.
Okay, let's pause and look at the grand accounting. We started with 6 carbons in our original glucose molecule way back in the lobby. We lost 2 carbons as CO2 during the link reactions, 1 for each pyruvate. And now we've lost 4 carbons in the Krebs cycle, since the cycle turns twice, once for each acetyl-CoA. 2 plus 4 equals 6. All 6 original carbons are gone.
All gone.
The physical structure of the glucose molecule had been completely and utterly dismantled into carbon dioxide gas.
That is a phenomenal milestone. The first crucial objective of aerobic respiration is complete. The stepwise, methodical removal of all hydrogen atoms and electrons, leaving only the oxidized carbon skeleton to float away as CO2. But the Krebs cycle itself isn't over.
We still have the 4-carbon piece left.
We are currently holding this 4-carbon Succinyl-CoA, and we must regenerate our starting material, the 4-carbon OAA, so the cycle is ready to receive the next acetyl-CoA.
And this regeneration phase contains a massive high-yield focus area. It happens during the very next step, the conversion of Succinyl-CoA to succinic acid.
Why is this specific conversion so heavily tested?
Because it is the one and only point in the entire Krebs cycle where substrate-level phosphorylation occurs.
Explain what that means mechanically.
Most of the energy we've harvested so far has been locked away in those NADH casino chips. But during this specific conversion from Succinyl-CoA to succinic acid, the bond breaking releases enough pure energy to directly forge a high-energy phosphate bond. It synthesizes a molecule of GTP.
Guanosine triphosphate.
Right. And in a rapid coupled reaction, that GTP immediately transfers its phosphate to an ADP molecule, creating a brand new molecule of ATP. Examiners love asking exactly where substrate-level phosphorylation happens in the TCA cycle. The answer is always the Succinyl-CoA to succinic acid step.
That is a classic exam trap. Examiners want to verify that you understand the fundamental difference between making a carrier molecule like NADH, which requires later processing, and directly synthesizing an immediate energy molecule like ATP or GTP right on the spot.
The cycle then rapidly moves through its final regeneration steps. The succinic acid is oxidized into malic acid, and finally malic acid is oxidized back into our starting molecule, oxaloacetic acid. During these final structural shifts, more high-energy electrons are harvested. We get one point where a different carrier, FAD plus S, is reduced to FADH2, and one final point where NAD plus is reduced to NADH.
Let's summarize the harvest of the Krebs cycle per single turn. From one acetyl-CoA entering the cycle, you extract 3 molecules of NADH, 1 molecule of FADH2, and 1 direct molecule of ATP. Since one glucose molecule provided 2 acetyl-CoAs, you double all those numbers for the total per glucose yield.
So if we stop and look around the mitochondrial matrix right now, the original glucose structure is completely vaporized.
Yep.
It's gone. And strangely, we haven't actually made very much usable ATP at all. We have a net of 2 from glycolysis and 2 from the Krebs cycle, 4 measly ATP.
That doesn't seem like enough.
What we do have, however, is a massive vault stacked to the ceiling with these electron carrier chips. 10 NADHs and 2 FADH2s.
Exactly. And that brings us to the ultimate pressing question of respiration. What on earth do we do with all these chips? Up to this point, oxygen hasn't even entered the biochemical picture, nor has the promised massive payload of ATP been synthesized. How does the cell extract the kinetic energy stored in these static electron carriers?
We have to take them to the cashier. We are entering part 5, the grand finale, the electron transport system, or ETS. And to do this, we are shifting our physical location inside the cell one last time.
Where are we going?
We're leaving the fluid of the mitochondrial matrix and moving directly onto the inner mitochondrial membrane itself.
And remember your anatomy. The inner mitochondrial membrane is heavily folded inward into structures called cristae, specifically to maximize the surface area. Embedded deep within this incredibly vast membrane is a physical series of giant protein complexes. This is the metabolic highway through which our high-energy electrons will be passed from one specialized carrier to another.
The text outlines these specific complexes, and I want to make sure we hit the precise high-yield details that examiners actually care about.
Let's walk the highway. We start at complex I.
Complex I is a massive enzyme called NADH dehydrogenase. Just like the name implies, this is the cashier's desk, where the NADH molecules from the matrix cash in. The NADH binds to the complex, is oxidized back into NAD plus I, and physically drops off its high-energy electrons.
And where do those highly reactive electrons go?
They are immediately transferred to a molecule called ubiquinone, which is a lipid-soluble carrier located right within the core of the inner membrane.
Now, what about the FADH2 molecules we generated in the Krebs cycle? Do they also drop their electrons at complex 1?
No, and that's another brilliant distinction. FADH2 is a slightly lower energy chip. It bypasses complex I entirely and drops its electrons off at complex 2, which is officially named succinate dehydrogenase. From complex 2, those electrons are also routed directly to ubiquinone.
So ubiquinone acts as a central collection pool in the membrane. Once it receives electrons from either complex I or complex 2, it becomes reduced to a state called ubiquinol. What is the next step on the highway?
The ubiquinol passes its electrons down the line to complex 3, the cytochrome BC1 complex. From there, the electrons need to jump to the final destination, complex 4. And they make this jump via a highly mobile carrier called cytochrome C.
Cytochrome C, very important.
The text describes cytochrome C very specifically. It is a small protein attached to the outer surface of the inner membrane, acting as a physical shuttle moving back and forth between complex 3 and complex 4.
That exact physical description, a small protein acting as a mobile carrier situated on the outer surface of the inner membrane, has been the verbatim text of countless multiple-choice questions. It is a vital spatial detail to remember. Now, we finally arrive at complex 4, the terminal complex. Cytochrome C oxidase. What is inside this final machine?
Complex 4 contains cytochromes A and A3, and here is the major exam trap I noticed. It contains 2 highly specific metallic centers. What is the element?
Here is what you must remember. Complex 4 relies on 2 copper centers, not iron, not magnesium, but copper. Examiners test this relentlessly. They will present a question asking which complex features copper centers, or they will list a jumble of components and ask you to identify the correct functional makeup of complex 4. You must inextricably link complex 4 with copper in your mind.
Copper centers in complex 4. Got it. Now, as these electrons are bouncing down this highway from complex to complex, they are moving to progressively lower energy states. But electrons don't just move on their own. Something has to be pulling them.
And this brings us to one of the most profound realizations in all of biology. We call this entire hour-long process aerobic respiration. But we've gone through the entirety of glycolysis, the link reaction, the Krebs cycle, and almost the entire length of the ETS highway, and oxygen hasn't done a single thing.
Not a single thing.
It hasn't participated in a single reaction until the very last millisecond of the entire process.
It's unbelievable. Oxygen's role is strictly, exclusively limited to the terminal stage. But its presence at that exact terminal stage is absolutely non-negotiably vital for everything else to work. Why?
Because oxygen is the ultimate, incredibly powerful garbage collector for the electrons. It is the final hydrogen and electron acceptor. Oxygen is highly electronegative. It sits at the very end of the transport chain and exerts a massive magnetic pull, dragging the electrons through the entire system.
So if oxygen wasn't sitting at the end of the line, the electrons in complex 4 would have nowhere to go. Complex 4 would stay full. Then cytochrome said he couldn't drop off its electrons, so it would stay full.
Right.
Then complex 3, then ubiquinone, then complex 1. The entire highway would instantly back up like a massive traffic jam.
Exactly.
The NADH wouldn't be able to drop off its electrons, the NAD plus wouldn't be regenerated, and the Krebs cycle would violently crash to a halt.
Oxygen prevents the traffic jam. It graciously accepts those tired, depleted electrons at the end of the chain, combines them with a couple of protons floating around, and forms a perfectly safe, stable molecule of metabolic water. Oxygen's only job is to clear the exhaust out of the tailpipe so the engine can keep running.
It's a breathtakingly elegant design.
Yeah.
But we are missing the final piece of the puzzle. The text notes that as electrons move down this chain, energy is released. But we still haven't actually phosphorylated anything yet. We haven't made the ATP. How does the physical movement of electrons down a membrane translate into the synthesis of ATP?
This is explained by the chemiosmotic hypothesis. As the incredibly high-energy electrons jump through complex 1, complex 3, and complex 4, those massive protein structures act like physical water pumps. They use the energy of the passing electrons to actively pump protons, hydrogen ions out of the matrix, across the inner membrane, and into the confined inner membrane space.
And what does pumping millions of positive protons into a tiny confined space accomplish?
It creates a massive, volatile electrochemical gradient. It is exactly like pumping millions of gallons of water up into a reservoir behind a hydroelectric dam. You are creating a vast store of potential energy. The protons are highly concentrated and positively charged. They violently repel each other and desperately want to flow back into the low-concentration matrix to balance the pressure.
But the lipid membrane is a solid wall. It is completely impermeable to them.
Except for one highly specific release valve.
Right. Complex V, ATP synthase.
ATP synthase.
I looked deep into the structure of ATP synthase, and it is a literal physical molecular machine. It has two major components. F0 is the integral membrane protein complex that acts as the physical pipe or channel through the dam, allowing the protons to shoot across. F1 is the peripheral headpiece protruding out into the matrix, and F1 is where the actual catalysis happens.
The visual of this is stunning. As the protons flow down their intense pressure gradient, shooting through the F0 channel, the physical force of their movement actually causes the internal stalk of the enzyme to physically rotate. It spins like a turbine at thousands of revolutions per minute.
That is crazy.
This violent mechanical rotation forces the F1 headpiece to undergo structural changes, literally slamming ADP and inorganic phosphate together with such force that they bond, creating ATP.
It is a microscopic rotary motor powering every living thing. And for the exams, we need to know the strict mathematical exchange rate of this turbine. The text states explicitly, for each molecule of ATP produced, exactly how many protons must pass through the F0 channel.
Here's what you must remember. Exactly 2H plus 92 protons must pass through F0 from the intermembrane space back to the matrix to synthesize 1 ATP. Memorize that ratio. 2 protons per 1 ATP.
And based on that proton pumping math, we finally get the exchange rates for our casino chips. The text tells us that the oxidation of one molecule NADH at complex I pumps enough protons to eventually spin the turbine enough to synthesize 3 molecules of ATP. The oxidation of one molecule of FADH2, because it enters later at complex 2 and pumps fewer protons, only produces 2 molecules of ATP.
We have finally seen the conclusion. The massive, dangerous energy of the original glucose molecule, which was carefully dismantled and temporarily held in the electron carriers, has finally been safely converted into a large payload of usable ATP, which naturally brings us to part 6, the balance sheet.
If we sit down and do the theoretical math, adding the 2 direct ATP from glycolysis, the 2 direct ATP from Krebs, plus the massive payout from cashing in all 10 NADH yielding 30 ATP, and 2 FADH2 yielding 4 ATP at the ETS, we arrive at a grand total net gain of 38 ATP molecules per single glucose molecule during aerobic respiration.
38 ATP. That is the classic textbook number. But we desperately need a reality check here. The text makes a specific point of highlighting the massive assumptions required to get this perfectly clean mathematical number. What are those assumptions?
To get exactly 38, we have to assume a perfect, orderly, uninterrupted pathway functioning sequentially. Glycolysis must finish entirely, then Krebs, then ETS, one after another like a factory line. We have to assume the NADH produced in the cytoplasm lobby is perfectly, flawlessly transported into the mitochondria without energy loss.
What else?
We have to assume that not a single one of the carbon intermediates in the entire pathway is ever siphoned off to build other cellular structures. Yeah. And we have to assume that absolutely nothing but pure glucose is being respired.
And do incredibly complex, chaotic living systems actually operate like perfect, closed mathematical models?
Not at all. The text explicitly warns that these assumptions are not really valid in a living biological system. In reality, all these pathways are working simultaneously. Substrates are constantly entering and being withdrawn. ATP is utilized the exact millisecond it is needed. It is a dynamic, swirling, deeply interconnected web of chemistry, not a straight, rigid assembly line.
Which perfectly sets up the final, most profound paradigm shift of this entire chapter, the concept of the amphibolic pathway.
This was the final concept that really made everything click for me. For this entire discussion, we have treated respiration purely as catabolism. We viewed the cell as a demolition site violently breaking down glucose into carbon dioxide just to extract energy. But that is only half the story of respiration.
How so? Explain the other half.
Because the living cell isn't just a demolition site. It is also a highly active manufacturing factory. It constantly needs to build new things, a process called anabolism. And it turns out the respiratory pathway isn't a one-way street to destruction. It is the central chemical hub for both breaking down and building up. Let's say the plant cell suddenly needs to synthesize new fatty acids to expand its cell membrane. It doesn't have to build them from scratch. It simply reaches into the Krebs cycle, withdraws a molecule of acetyl-CoA, and uses that as the pre-built foundational block to construct the fat.
It acts as a warehouse of parts. And conversely, if the cell is starving for energy and only has stored fats available, it breaks those fats down, the fat is cleaved into glycerol and fatty acids, the glycerol is modified and enters the respiration pathway directly as PGAL in glycolysis. The fatty acids are degraded into acetyl-CoA and dropped directly into the Krebs cycle.
It works for proteins, too. If the cell is utilizing protein for fuel, proteases break the long protein chains down into individual amino acids. After a process called deamination removes the nitrogen, those carbon skeletons can enter the pathway at various points depending on their specific chemical structure. Some enter as pyruvate, some as the acetyl-CoA, and some drop straight into the intermediate stages of the Krebs cycle itself.
Because the respiratory pathway is so deeply involved in both the catabolic breaking down of molecules for energy and the anabolic withdrawing of intermediates to synthesize entirely new molecules, it is biologically inaccurate to simply label it a catabolic pathway.
Here is the absolute key term you must remember for this concept. It is an amphibolic pathway, amphi meaning both. Just like an amphibian lives in both water and land, this pathway exists in both the realms of catabolism and anabolism. It is a dynamic, two-way street of metabolism.
Beautifully explained. The final quantitative concept we need to cover today, which ties all these different fuel sources together, is the respiratory quotient, or RQ.
Right. The RQ is a simple mathematical ratio. It is the exact volume of carbon dioxide gas evolved, divided by the volume of oxygen gas consumed during the process of respiration.
Why do examiners and biologists care so deeply about the RQ? Because the value of the RQ is a perfect diagnostic tool. It tells you exactly what kind of fuel the cell is currently burning. You must memorize these three baseline numbers.
Let's run through them. When pure carbohydrates, like our standard glucose molecule, are used as the substrate and completely oxidized, the RQ is exactly 1. And the math is simple. The chemical equation is C6H12O6 plus 6 molecules of O2 yields 6 molecules of CO2 plus 6 molecules of H2O. 6 molecules of CO2 evolved divided by 6 molecules of O2 consumed equals exactly 1.
What happens to the ratio if the cell switches its metabolic engine to burn fats?
For fats, the text uses a specific lipid called tripalmitin as an example. When burning fats, the RQ is significantly lower. It drops down to roughly 0.7.
And why is it lower, chemically speaking?
Because fat molecules are highly reduced. They contain very little oxygen inherently in their massive hydrocarbon chains compared to sugars. Therefore, the cell has to pull in a vastly larger amount of environmental oxygen to completely oxidize that fat relative to the amount of carbon dioxide it eventually gives off. More oxygen consumed in the denominator makes the final ratio smaller.
Spot on. And for proteins?
If proteins are being forced into use as the respiratory substrate, the RQ ratio sits roughly at 0.9.
So lock these tiers into your memory. Carbohydrates equal 1, proteins equal roughly 0.9, fats equal roughly 0.7. But always keep in mind the text's final realistic caveat. In actual living organisms, respiratory substrates are almost always a complex mixture of more than one type at a time, and pure proteins or pure fats are almost never used in total isolation as the sole substrate.
We have covered an immense amount of ground today. We traveled from the cytoplasm lobby, mapped the investment and payoff of glycolysis, witnessed the desperate recycling of fermentation, entered the VIP matrix for the Krebs cycle countdown, and rode the electron highway to the spinning turbine of ATP synthase. Before we wrap up, could you give the listener that 60-second sticky summary The absolute non-negotiables they need to take into the exam.
Here is your rapid-fire synthesis. 1. Glycolysis happens entirely in the cytoplasm. It requires 0 oxygen. It nets 2 ATP, 2 pyruvate, and 2 NADH. The key trap is the enzyme invertase for breaking down sucrose, and remember that NADH is minted specifically at the PGAL to BPGA step.
Got it.
2. Fermentation is a desperate recycling loop meant only to regenerate NAD, plus so glycolysis doesn't stall. It yields terrible energy, creates toxic byproducts, and relies on alcohol dehydrogenase or lactate dehydrogenase.
Alcohol and lactate.
3. The link reaction connects the two worlds. Pyruvate becomes acetyl-CoA. It absolutely requires pyruvate dehydrogenase and the heavily tested magnesium ion trap, Mg2 plus Covin-4. The Krebs cycle operates in the mitochondrial matrix. It completes the total carbon breakdown, yielding the vast bulk of your NADH and FADH2 chips. Look out for the single GTP substrate-level phosphorylation trap at the succinyl-CoA step.
That succinyl-CoA step is huge.
5. The ETS is located on the inner membrane. Complex 4V has the famous copper centers. Oxygen acts as the ultimate garbage collector for electrons, pulling them down the chain to make water, while the ATP synthase turbine uses the massive proton gradient to churn out the bulk of the ATP. And finally, because it handles both destruction and construction, respiration is an amphibolic pathway.
That is the perfect mental map. As we wrap up this deep dive, I want to leave you, the listener, with one final provocative thought. We just spent this entire time outlining the excruciatingly specific enzymatic pathway that keeps a microscopic plant cell alive. But I want you to consider the sheer scale of what we just learned. The specific enzymes of glycolysis operating in the cells of the green oak tree outside your window right now are functionally identical to the ones operating inside your own human muscle cells as you sit here listening.
Unbroken chain.
And they are identical to the enzymes functioning in the ancient anaerobic bacteria that lived in the dark primordial oceans billions of years ago, long before the Earth even had a wisp of oxygen in its atmosphere. When you study respiration, you aren't just memorizing abstract chemistry for a test score. You are literally looking at the unbroken shared engine of all life on Earth. You are tracing a continuous microscopic chain of energy transfer that stretches back to the very dawn of biology. How is that for a connection?
It truly puts the entire subject and biology itself into a breathtaking perspective.
It really does. Thank you so much for taking this deep dive into plant physiology with us. Keep studying, keep looking for the why behind the what, and keep seeing the big picture. We will catch you on the next deep dive.